you for having me, and I'm looking forward to sharing my work and also hearing the questions that people have. So I'm going to start by sharing my screen and can you see the screen?
Yes.
Okay, great. Awesome. Well, it's great to virtually meet everybody. And today I'm going to talk to you a little bit about the work I've been doing over the past, I guess, 10 years at this point on what biological robots can teach us about fighting disease. And as I mentioned, or as was mentioned in the introduction, I'm currently a postdoc at MIT, and I've put my contact info here and at the end of the slide as well. So I know that I don't come up with questions right away. Sometimes it comes to me like 3 days later in the shower. So like if you're that kind of person, feel free to reach out on social media, DM me a question. I'll be happy to answer them. All right, so just to kick off, I wanted to start with a little bit of a personal introduction because I think it's nice to know kind of the story behind the science and why somebody might be inspired to do this kind of work. For me, that inspiration comes from my personal history with my family as well as my childhood. So I was actually born in India. I was born in Chennai, and I lived there on and off throughout the years in like elementary school, but I also spent a good part of my childhood in Kenya, and then I moved to the US when I was around 10 years old, and I've lived there ever since, moving around a lot here too. So I've moved around to all these other places, and the only kind of consistency in my life came from my family. So my mom, my dad, and my grandfather we're all engineers. My mom's a chemical engineer, my dad's a mechanical engineer, and my granddad was a civil engineer. And kind of, they've been like a really strong inspiration to me because I was always seeing how they were using their training to ask questions about their environment, to identify problems in their environments. And they always seemed to have the tools required to build a solution to a problem that they were facing in their life. And so I think that sort of sense of agency that I saw in them was really inspiring to me. And always, even though I didn't always know that I wanted to be a scientist or an engineer, I knew that that was a path that could lead to some really interesting things in my life. For example, one particular story that I will highlight is that when I was growing up in Kenya, we lived most of the time in Nairobi, which is a city, but we also spent a lot of our weekends at villages, at rural villages in Kenya, where my dad's company was helping put up communication towers that would connect the villages to the rest of the world. And this was, I think, a really, really powerful demonstration for me that showed that, you know, with this mechanical engineering degree and training, you could do something that was really impactful for a community and that would have lasting change, and that you could share that knowledge with a lot of other people. So, you know, these are the kinds of things that happened to me in my early life that convinced me to apply for a mechanical engineering program at Cornell University, where I did my undergrad. And when I was there, I was like, I'm gonna be a mechanical engineer because I wanted to be an astronaut at that time, because who doesn't? Everyone wants to go to space, right? Or I think most people. So I'm like, all right, I'm going to be an astronaut. So I signed up for the mechanical and aerospace engineering degree program at Cornell. But for some reason, I couldn't figure out how to sign up for the Intro to Aerospace class. So I ended up in an Intro to Biomedical Engineering class because a girl who lived next door to me in my dorm said that the professor who taught it had a really great British accent. I'm like, well, that sounds like a good enough reason to take a class as compared to anything else. So I take the class, and it turns out that in addition to having a great accent and being just a very funny and engaging educator, he also just was able to convey the excitement of this field and talk about how engineers actually had a lot to contribute to medicine, both in understanding why diseases occur and also developing strategies to treat them when they do occur. So that really opened up the possibilities for me of me being able to do something for medicine. But I don't think I really saw my path into that field until my first job. So my first job or internship, unpaid, so like not a real job, but you know, it was something, it was experience, was at in a lab, a biology lab, where they were studying skeletal muscle, and they were specifically looking at the fact that people who exercise a lot, we know, have really strong muscle and like really strong muscle tone, but people who drink a lot of alcohol, like abusively, can have their skeletal muscle degenerate. And so they're like, what if you do both of those things? What if you drink a lot and also work out a lot? Can those two things kind of cancel out and essentially help out your muscle, at least achieve some sort of baseline? And so they needed somebody to get rats drunk and put them on a treadmill and watch them run and look at how their muscle tone and their weight and their behavior was changing over an extended period of time. And that might sound a little bit ridiculous, but that's actually one of the strongest ways that we had at that time to test a hypothesis like that. So, of course, I was the poor soul that was stuck in a basement of the lab, putting these rats on treadmills and watching them run. And it was kind of depressing, like for just to sit there for hours and watch this. But over time, I guess I had a lot of time to think and meditate about what was happening. And I realized that what was really interesting about these rats and about the muscle that was powering their running is that it was dynamically changing in response to its environment, right? And like, we were working with all these actuators in our classes, in my mechanical engineering classes, motors and shape memory alloys and whatever it is. And all of those things were working great, but they weren't responding to their environment in any way. So there was something really special about muscle, and that stood out to me. And it kind of brought on this broader philosophical question of, for thousands of years, engineers have been building with things like wood and metal and plastics, but that doesn't really help us in the same way that our bodies help us. Those materials, I could pick up my phone right now and I could smash it against the ground, right? And I could crack the screen. And then what happens? Like, I'm going to have to go get it replaced, or I might need to even buy a new phone. There's nothing that that screen can do. But if I fall off my chair right now, which happens a lot, it's kind of a wobbly chair, and I scrape my skin or I break my bones, my body is able to react to that in some way and help me achieve the function of my skin and bones that I had before, or at least some approximation of that. So there's something clearly interesting and unique about biological materials where they're able to dynamically sense and respond to their environmental signals in ways that the traditional materials we build with don't do. And that really raises the question of what can we build with biological materials? And if we built, say, machines made out of these, would they be able to self-assemble or self-heal in ways that our bodies and other biological systems can do? Of course, now to be able to do that, you actually need a tool. You can just say, I want to build with biology, but or biological materials, but like, how are you actually going to do that? And for me, that answer came in the form of 3D printing. There are a range of tools that you can deploy and a range of different types of 3D printers that you can deploy. But I focused on a specific kind of 3D printing called stereolithography, where you use light to turn a liquid resin that is photosensitive into a solid. And so we would I showed that you could pattern cells and biomaterials and proteins in these complex 3D structures to create different types of shapes, essentially, with living cells embedded inside. So the obvious application of these kinds of technologies is in what we call reverse engineering, or creating tissues or organs like the heart that's shown on the left of the screen here that look exactly like tissues or organs inside our body. The idea being that if we were able to do this at scale and in a way that really replicated the structure in our bodies, then if you got sick, if you had a heart attack, if you lost an arm in an accident, then maybe one day we would be able to replace it with these 3D printed replacements. And now we're obviously very far from that, but a lot of people are working on the beginning aspects of that problem in this reverse engineering space. But because I was a mechanical engineer, and really just interested in design and machinery, I was like, well, that's really great. But what can we do with biological materials that doesn't mimic what already exists in nature? Can we build machines or robots, for example, that forward engineer or use biological materials in new ways to do things that don't already exist in nature? And so that concept of forward engineering became very interesting to me and ended up what I focused my PhD on at the University of Illinois. All right. So I ended up at the University of Illinois as a mechanical engineering PhD student, and I ended up pursuing research at a center with the National Science Foundation between Illinois, MIT, and Georgia Tech, as well as a range of other partner institutions. And this whole center was focused on this idea of biological machinery because obviously I'm not the first person that thought of that or the only person that thought of that. And as a whole goal, our center was to say, can we make machines powered by biological robots or powered by biological materials, and specifically robots as a class of machines? So just for the sake of having a definition that we're all on board with and all working with, I define a robot, I think a lot of people define a robot as a device that somewhat autonomously senses, processes, and responds to signals in its environment in real time, whatever that real time can be. And a biological robot or biobot or biorobot is a robot that uses biological materials to perform one or all of those functions, either sensing or processing or response or all three. And the goal of building these kinds of robots would be the same thing as, the advantages that biological materials have over synthetic materials. So we can say, if I made a robot that it had biological components, would it be able to do some of these adaptive functional behaviors like self-healing that we can't currently see in robots that we currently have with synthetic materials? And so as a goal, as a center, we were like, can we make, a robot that senses some toxin, moves towards it, and neutralizes that toxin? And that's a huge, huge fundamental technical problem. And so we all decided to kind of break that up into pieces and focus on specific aspects. And I was charged with the problem of like, can you get the robot to move and walk around? So specifically focused on actuation. So when I started my PhD, in 2012, there were actually a couple great examples of people that had already made robots that moved and walked around powered by biological materials. And I'm showing you videos of both here that I hope you can see. On the left is a swimming jellyfish-like robot, and on the right is a walking sort of inchworm-like robot. And both of them kind of operate on the same principle, which is that they're made of a flexible polymer on top of which has been seated a layer of cardiac muscle cells. And when all the cardiac muscle cells beat, the polymer deforms, and so you're able to create these kinds of motions. So they focus on motion because it's a really intuitive demonstration of functionality, but also pretty much every machine needs to generate force and produce motion. So it's something that's really important and broadly applicable. And they pick cardiac cells or muscle cells from the heart because these cells produce a lot of force. Obviously, they're pumping blood throughout our bodies. They don't need you to tell them to turn on or off. They're just beating all the time. Also makes sense. Your heart never turns off. And if it does, you're in trouble, right? And they also all beat at the same time, which also makes sense. All these cells are electrically connected to each other. So they beat at the same time so that our heart is functioning as a pump. If they were beating at different times, you would have an arrhythmia and you might have, again, might end up with problems. So it makes sense that they used cardiac muscle, but there are some limitations of this. And part of it is that lack of on-off control. Those of you who are engineers or aspiring engineers might realize that having a machine that never turns off is not necessarily something that's super advantageous. And also, heart muscle cells have limited regenerative capacity. They can't necessarily heal after damage. This is why things like heart attacks end up being such big problems. So you want something ideally that is robustly resilient to damage, and that led me to think about skeletal muscle, because that really kind of addresses a lot of the problems we have. So skeletal muscle in our bodies is what powers all of our voluntary motions, right? Like moving, dancing, running, all of those things. It can heal after small damages. It can be turned on or off through electrical signaling, usually from motor neurons in our peripheral nervous system. And it's also just a really good actuator in the sense that if you compare the force per unit volume, it does much better than most synthetic actuators that we have to date. And so it has this really nice hierarchical structure where at the microscale, there are these two protein filaments, actin and myosin, that are sliding against each other. But at the macroscale, when this happens several times in series, you can see these really large contractions. So as an actuator, you can see why it might be a very appealing tissue of choice for people to use. But how do you actually go from saying like, okay, I understand that muscle produces force to here is a robot that is powered by muscle? And I think that's where kind of taking inspiration from biology really comes to mind. So in our bodies, skeletal muscle is tethered to our bones via relatively stiff tendons and then stretched across an articulating joint such that when the muscle contracts, you can generate these sort of large locomotions or actuation behaviors. So you can recreate that in the lab by first looking at that sort of skeletal structure and saying, how can I do something that looks like that? So we used a 3D printer to print a low stiffness beam that mimicked that articulating joint, at the ends of which we put these high stiffness pillars that mimic the tendon. And then we said, if we tethered the muscle to these two tendons, we should be able to generate something that can contract and deform the overall structure. And to actually create that muscle, we took some living cells, mouse skeletal muscle cells, mixed them with a whole bunch of proteins. And so it kind of looks like this jelly or goop like solution. We injection molded around this skeleton. And over time, because they're living, the cells self assemble. So they proliferate, they exert forces on their environment, they're compacting that tissue around themselves, and they fuse together to form these long fibers that are these green lines shown in this picture here. And that is the basic contractile unit of skeletal muscle. So once it gets to those long fibers with multiple cells fused together, you know that it should be able to start generating force and producing motion. So already you start seeing that even without contraction or anything, the muscle is generating force. It generates a passive tension force that is counteracted by the skeleton that you've printed the muscle around. And you start seeing that it starts responding to its environment, which is really interesting because with any kind of synthetic material or actuator, you know, you say produce this much force, it produces that much force. That's all it does. It doesn't respond to its environment in any way. It can't change its behavior after you've already put it inside the machine. But muscle can. If you print it, if you pattern it around a stiffer skeleton, it can generate larger forces because it's getting more aligned, it's producing different kinds of proteins, it's maturing at a higher rate. If you put different growth factors into the media, that's the solution that the muscle is sitting in, that's feeding it, you see that it can dramatically increase force production as well. And so already you start seeing that building with this biological material has started making a difference. The robot is responding to its environment in a way that a traditional robot wouldn't do. But of course, that's interesting, but what you're actually interested in is measuring muscle contraction, right? So the way we do that is, as I mentioned before, in the body, the muscle is getting signals from neurons that are telling it to contract, and we don't have that in this system. But one thing that we can do is place it between two electrodes and send electrical pulses through the tissue such that we can elicit a contraction. So if you send a pulse at like 1 cycle per second or 1 hertz, you get one contraction. If you do it at 4 cycles per second, you get four contractions. One thing that you do see is that it does change a little bit over time, in that if you're doing 1 hertz contraction, the muscle can contract and then relax completely before you tell it to contract again. But as you get to these higher frequency contractions, the muscle can't fully relax before it can contract again. So you can start seeing that the dynamic range of the force that's being produced gets smaller. And this is actually something that's very similar to what happens in natural muscle. So we know the muscle is behaving as it should. The next thing that we did is looked at, can we get these robots to actually walk? It's cool that they're producing force, but can we get them to actually do something? So the way we did this is by first doing some little computational modeling to say, oh, like if a symmetric structure exists and we contract symmetrically, we're going to get something that looks like dancing, but it's not really going to do much, right? However, if you make the structure slightly asymmetric, where one leg is a little bit longer than the other, you can get the robot to slowly walk in the direction of the longer leg. So that was our prediction, and actually what we ended up seeing, as hopefully you can see in this video, the left leg is a little bit longer, and so with every muscle contraction, the robot is slowly crawling in the direction of the left leg. And another thing that we saw that was very interesting is that for slightly higher frequency stimulations, we started seeing slightly higher frequency or slightly higher speeds of walking, which makes sense as well. So we can make the robots walk, which was great. It was actually the first demonstration of robots powered by engineered skeletal muscle that could walk. And it's always exciting to be the first one to do something, right? But also when you're working on something, you see the flaws more than anybody else. So one flaw that I saw was that one, these robots can only walk, and two, they can only walk in one direction. What use is that? So the way that I fixed it is by one, genetically engineering the cells so that they would be responsive to blue light. And so that means every time we flash blue light on them, they would contract, which means that you can focus the blue light at different parts of the tissue, right? So you don't have to activate all of the muscle at the same time as with electrical stimulation. And I thought maybe this is a way we can get it to do more interesting behaviors. And the second thing we did is we converted to a more modular design. So rather than patterning the cells directly around the muscle muscle itself, we patterned it into a ring or rubber band-like structure, which we could then pick up and transfer to any of a wide variety of skeletons. So there's this video here of me picking it up and transferring it to one of our sort of walking design skeletons that I showed you before, but other people have picked up these rubber rings and put them around a tube and tried to make a peristaltic pump, for example. So there's a lot of different types of designs and ideas that you can pursue by making this relatively simple modular change. I hope this video also kind of gives you a sense of the scale of these robots. There's several millimeters long. We've made some that are in the centimeter scale, but those are my hands. So you can see that they're quite tiny. All right, so then again, in these kinds of ring-like robots, we start seeing that the internal structure mimics natural muscle and also mimics its environment. It responds to its environment. So when we put these cells in the mixture itself, they're not in any particular order. They just sort of fuse together, right? But when they fuse together into these fibers, there's nothing technically that we are telling them beforehand, like you all have to align in the same direction and produce force in the same direction, but they can sense it. They can sense the mechanical environment of the skeleton that's pulling them in this direction. And so when they experience these tension forces in this central area, you can see that the myotubes are highly aligned along the axis of tension. And so when each of them contracts, they're actually all contracting in the same direction, which means that that's why we're seeing macroscale contraction in these systems. If they were all acting in different directions, you might not really see anything. So again, you start seeing that it's responding to its environment, but of course you want to actually see that you are able to elicit contraction from them in some way. So instead of using electrical stimuli this time, we used an optical stimulus because we had genetically engineered these cells, right? So they should be able to contract when they get a light signal. So we put an LED on top of them and just kind of flashlight, kind of like a disco ball, and saw what happened. And we saw, in fact, that every time we flashed a pulse of light on them, the muscle would contract and deform the skeleton. And by knowing the mechanical properties of that skeleton and the size of it, we could back calculate the force that was actually being generated. So we did that and again repeated some of our experiments from before. Again, if you have a robot that has a leg that's a little bit longer on the right side, you can see that the robot is slowly crawling in the direction of the longer leg, and that the speed of the robot can be regulated by increasing the frequency of contraction. But of course, this is just a repeat of what we did before, and we want to do something a little bit different, right? The whole point of using light is to say that you don't have to make the structure asymmetric. You just need to make the muscle contraction asymmetric. And so what we did was make these completely symmetric two-legged robots and showed that if we only excited one of the legs with light, or if we only excited half of the legs with light, we could do things like get them to walk in different directions or move and turn around. So hopefully what you can see in these videos is that we have robots that walk in one direction preferentially because they are following the light. And in the other case, in the right-hand video, you can see that the robot is slowly rotating, again, because it's following the light. Only the muscle that is being excited by light is contracting. And so you can essentially get a robot to navigate 2D space. And we were able to do this fairly consistently across a lot of different robots, so we were confident this was something repeatable. And the last thing I want to share with you about the space is, I started with like, what can these robots teach us about disease? And I think that's kind of where the end of my PhD has really focused, partially because I was like, well, I made some robots that walk, but we already have robots that walk. I haven't proved that biological robots can do something that synthetic robots can't do, right? And so I started this talk and my motivation for this research by talking about healing, how our skin can heal when our phone screens can't heal. And I decided to focus the end of my research during my PhD on that as well. And thinking about the fact that if I can show that these robots can heal, then I would really have proved that there's an advantage to building with biological materials. So first thing we did is actually we had to go in and damage them. So we made the robots the same way as before. We go in and we create a tear in the muscle, and that tear over time propagates and the whole muscle rips apart and it's done. Like, you can't do anything. And the larger tears result in larger drops in the force that's being produced, and the muscle cells are dead. And this makes sense. This is exactly what should happen because those long fibers are something that we call terminally mitotic, which means that they can't differentiate anymore. The way our body deals with this and heals that damage is that outside of the muscle, we have these small populations of stem cells or satellite cells that sit around the muscle. And when they sense damage, they move to the site of damage and proliferate. They create more cells and then they fuse together to form new fibers. So we realized that we would need to replicate that in our system. So that's what we did. We went in and first tested, like, if I just heal this tear with some sort of biological glue, is that enough to recover force? And turns out it wasn't. Those cells were dead and then we weren't recovering them in any way. But in the glue, when we mixed in some new cells, some muscle stem cells that hadn't formed these long fibers yet, then we saw that slowly we were able to recover force after 7 days post damage. But you can see that the force that's being recovered is still less than the force that was being produced before damage, right? Um, but turns out that muscle can still do a lot of really interesting things. So we learned that if we exercise the muscle for 30 minutes with 1 hertz blue light stimulation every day, and also did local release of growth factors that promote muscle health and maturation, we could actually completely recover the force that was being produced by the muscle after 2 days post damage. So you can see in this fluorescent image here, this red tissue is the original muscle. And the green tissue are the newly added cells that we included with the glue that we put in. So over time, you can see that the cells fuse together, they form these long fibers, they extend across the gap, they essentially bridge that gap, and they're able to generate force and produce motion at the same level as before. And so this was, I think, the real showstopper for me because there have been no demonstrations of synthetic actuators or robots to date that after they're broken can be revived in the same kind of way. If they're done and they have to be replaced. And so this was kind of the start of sort of what has become my line of research is saying, oh, look, like we can use, one, we've proven that biological robots can do things that synthetic robots can't do. But two, we can also use these robots as models of disease. We can look at how they respond to damage and we can develop new therapeutic strategies to combat that damage, which might be helpful if you lose, say, a large chunk of muscle. In an accident or through something else, maybe we can deploy those strategies in human beings that we learn in these robots. And so that's kind of what I'm working on right now. One is using these robots to ask and answer questions about human disease, whether it's something like damage, like I showed you before, or whether it's, say, a genetic disease where your cells aren't able to produce as much of a specific protein and you have a muscular dystrophy that affects you through the rest of your life. We can use these kinds of robots as test platforms for new gene therapies or other types of medicines that could potentially, if they have a curative impact on these robots, then we can study the mechanism of that and feel a little bit more comfortable choosing the therapies that we would want to eventually deploy in humans in clinical trials. The next scientific step that I also want to take is start thinking about the fact that, like, at this point you can just turn the muscle on or off. But in our bodies, our muscles are much more complex than that. They are being told to turn on or off by motor neurons, which are actually getting a lot of feedback from sensory neurons that are sensing stretch in our muscle, or might be sensing pain or heat from our skin. So it's a very complex feedback control loop that's not accounted for in the system right now. And so integrating those neurons and that sort of neuronal control will allow us to have a better understanding of muscle in our bodies, and also make robots that can navigate complex environments or serve their function with a greater degree of autonomy. They can do more than just on or off. They can respond to specific stimuli, process them, and make some sort of decision behavior. So that's kind of the goal. And eventually, over the long term, I think that we would be able to, deploy these robots perhaps as functional implants inside humans. So you can imagine that if we were able to create this muscle at a much larger scale, we would be able to use that to replace muscle that had been lost in the body, or perhaps serve as a factory of chemical factors that could help communicate with tissues around their systems and help us restore any kind of disease or damage that we were suffering from. So I know I threw a lot of information at you, and I'm happy to answer any questions that you have. I do want to wrap up by saying that we are working on a lot of things where we are teaching students at high schools, undergrad classes, how to build their own biological robots. And if you're interested in learning more, again, you can contact me on social media, but I also have a book coming out on this topic and other things that we can build with biology in September. So with that, I'd just like to thank all of the professors and labs and funding sources that have supported this work throughout the years. And I will keep this slide up as I answer any questions that you have, so you have access to my contact.
Thank you so much, Dr. Raman. That was very, very insightful. I think biorobots, this is a topic that we don't often hear about on an everyday basis. I mean, robotics, this is a topic that everybody has heard a lot about in the recent few years, but biorobots in particular, I think very, very fascinating for me. And I do think that the use of so many visuals that you incorporated in your presentation, it was very helpful to relate the kind of relate with the research that you were doing. So thank you so much for taking the efforts of putting everything together. We have received quite a few questions on Zoom as well as on social media where we're live streaming our events. So I'm just going to start asking you one question after the other. So the first question that I'd like to pose to you is, are there any ethical issues that you're dealing with while building biorobots? How are you dealing with this issue?
Yeah, that's a great question, and I'm so glad that you asked. So I think that there are definitely ethical issues in the sense that one big question that we ask ourselves, not only us, but anyone working with biological materials, is just because you're working with living cells, are you working with something that is alive? And that's a very philosophical question, right? Because you have to define what your definition of an alive thing is, what an organism is. Does that involve something that has some degree of autonomy or consciousness? And especially when we think about integrating neurons or nerves, there's one thing to be said for a nerve that can just sense a stimulus and respond to it in a certain way. And there's something else to be said that if we created a network of nerves that could resemble something that looks like the central nervous system or our brain, are we creating something that has a consciousness and can feel pain? So that's certainly something that we think about very proactively. And one way, my goal as a scientist is only to do things that are helpful to other people. And I really don't want to ever talk down to somebody and be like, you don't understand the science. Like, what do you know? This is fine. They're not alive. Like, I can drop them on the floor and they're good. And so I would say that right now, we're all in agreement. The scientific community and also sort of broader policymakers that we've engaged with are in agreement that these are not close to anything that can be resembled a living creature because they can't really autonomously metabolize their food in any way. They can't reproduce. They have no consciousness. However, if we did get to a stage where 50 years from now or 60 years from now, we were getting ever closer to creating something that looked more similar to a brain or was capable of decision making, then I think you absolutely have to be asking those questions. And I don't think you need to wait 50 years from now. So one thing that we try to do is I host these sort of bioethics discussions. I should have put a link in here of bioethics website that we have where we talk about different scenarios and we elicit feedback not only from other scientists but from the general public, philosophers. I work with a philosopher fairly regularly actually, policymakers, everybody, to get your feedback on like, when is the line where most people think it's too far, and then I just don't do that. And that's how I approach this work, and I think a lot of scientists approach this work as well.
Well, thank you. That was very well summarized. Like you correctly, I mean, you very beautifully put it across in the way that medical ethics, bioethics, this is a topic that scientists need to keep in mind on an everyday basis as we advance ahead in technology. And well, it's very nice to know that you and your colleagues are hosting symposiums on this topic, which many people need to hear about. We have linked your website as well as a couple of your social media handles in the chat. So I would encourage everybody to just take a look at the chat and make sure that you check out Dr. Raman's work online. Okay, so the next question that I have for you here is, so is there the work that happens inside our muscles, for example, lactic acid production, how are factors like these affecting the robots inside the body? How would they respond?
Yeah, so that's a really great question as well. And it's similar in the sense that the robot, the muscle, is metabolizing things in the liquid media surrounding it. It's creating ATP. It's using sugar in the media. It's converting it to ATP. It's having metabolic byproducts. It's different in the sense that because it's sitting in a liquid bath of media, a lot of that is just kind of draining out of the system, and we change that media every day. So it's not necessarily something where we have to create an active metabolic or vascular system where those waste products would need to be removed. However, if we were ever to make a robot that we were deploying, say outside of the construct of like sitting in a biosafety hood in a lab, then absolutely we would need to think about how do we metabolize those byproducts and how do we create all of the surrounding support systems that keep us alive in our bodies in these sort of robots. And even if we were going to say not deploy them into the world but implant them inside of our own muscles, we would need to think about how those chemical byproducts are affecting other tissues. And that's part of what I'm studying now. I developed this tool while I was at MIT for sampling the fluid between cells in sort of really complex tissues like the brain or in neuromuscular junctions. And we're looking at, in different scenarios, how does that interstitial fluid or the fluid between cells, how does its composition change? And how can we control that change? And I think this is one of those questions that we'll be asking and hopefully answering with that tool.
That brings me, I mean, you've made my job very easier because that links very perfectly to the next question that we have. How do you see biorobots playing out in the next 5 years? So you say that, we're right now we're in the research phase. When do you see it actually getting implemented into healthcare on a regular basis?
Yeah. So I think that's a great question. And also for people, aspiring scientists, I think it's really important to know that you can make a lot of important advances and it might still not get to humans for like 80 years. And that's the kind of sacrifice that you make when you're working at the sort of pioneering side of research. And so for me, I would say in the next 5 years, it is absolutely possible that we could use these muscles as models of disease and perhaps use them as a test platform to test therapies, which would then be deployed in animal trials, which would then be deployed in human clinical trials. In 50 years, I would definitely hope that this and other types of cell-based therapies would be regularly deployed in humans and could be used to fight disease and damage. And I hope that you don't get discouraged by that. I mean, one story that I like to share is that the person I work for, my advisor, Bob Langer, he's the co-founder of Moderna, which is one of the companies that has made a COVID vaccine. And research on that project in our lab started 40 years ago. And like, yes, it's helping billions of people today and is an amazing scientific product, but it's something that took people a very long time to persevere and work towards. And so that's kind of what I look towards is what can I get out of the next 5 years? I definitely ask myself that question, but I also think about what am I building towards over the long term in terms of how we can fight disease and damage. And I would love to hope that I'm working towards something that is as impactful as other researchers in my lab have worked on.
That definitely makes a lot of sense. Research isn't a 1 year or a 2 year affair. It goes on for many years, and you really need the perseverance to stand through and make sure that the research that you're putting in, have faith in the fact that this research will be useful for humans, say 40-50 years from now.
Yeah. And there are a
On
lot of wins along the way, I should say. Like every experiment that works is still exciting and still meaningful and moves science forward.
Definitely, absolutely. So the next question that we have here is, how is the biorobot going to be powered? So is it going to be battery driven? Is it energy driven? Can we look at sustainable methods of energy to power it? How exactly will it sustain in the body and so on?
That's a perfect question. And I think it's really important. I focus on health, but obviously a big challenge that we're facing in the world is not just in health, but also in energy and sustainable sources of energy. So I think that's a great question. And actually, one of my driving motivations for building with biological materials is that very thing. You don't need a battery to power muscle because it is powered by sugar, right? Sugars and amino acids in its environment are its battery. Now, you can make an argument that perhaps manufacturing sugar and amino acids at that scale might prove to not be as carbon neutral as we think it is, but it is possible. And so these are the kinds of questions we have to ask also, like what are the energy costs of using that sugar and amino acid to power the muscle? And even if they're high, is the fact that they're biodegradable something that we should look at as a positive? So definitely a great question. So I would say that our batteries are going to be these sorts of biological materials. However, I should say that energy and control are not the same thing. So if I were to continue using light or LEDs to control when it turned on and off, that would need something that looked more like a traditional battery, right? And so that's why when we think about can we integrate neurons, that's an additional component of this as well, because then you move away from that external battery-driven control towards something that is also powered by other biological materials.
Absolutely. That was really insightful. I think the next question that we have here is, so what are the precautionary measures that you have taken into account while working on biorobots in your lab?
Yeah, so we work with biological robots in very controlled environments. So because they're made out of cells that are derived from mice, so from mammals, they're actually very finicky about when they're going to work, which is they need to be kept at 37 degrees C at a specific humidity with specific oxygen and carbon dioxide. They can't get infected by bacteria or viruses, or like you can't drop them on the ground. Like they're going to die. Pretty much most of the time, they're going to die. So actually keeping them alive is the bigger concern. So we keep them, for the most part, we store them in these incubators that keep them at the right temperature, pH, humidity, air levels. And then when we actually need to do stuff with them, like when you saw my hands messing with them in the lab, that actually happens inside something that's called a biosafety hood, which is has sterile laminar airflow inside it. And so I have my gloved hands that are completely sprayed down with ethanol, using them inside there, taking videos, doing whatever I need to do, and then I move them back to the incubator. Now, if I took them out of that controlled environment for even, like an hour, they would die because the temperature is wrong, or they get too acidic, or they get bacteria, and we try to put like penicillin and streptomycin, wow, it's hard to say, in the media to prevent that from happening, but it does happen. And so when we think about in the lab, that's totally fine, and we're not necessarily concerned about it being able to escape that environment or do anything that we're not worried about. However, when you deploy it in another context, say inside the body, you don't necessarily have to worry about it dying because it should technically be at the right temperature and have all the biochemicals needed to survive. But you might want to start thinking about, are there off-target effects? And again, this is why it takes 50 years, 100 years to do these kinds of work, because you need to take very baby steps to say, like, if I just put the cell in an animal and then eventually a human for 1 day or 1 month, what happens? And if nothing happens, then we can start building that out a little bit further. But we purposely take very cautious steps to ensure that nothing bad does happen.
Oh, yeah, absolutely. Because this kind of research, I mean, research mostly involves working in very controlled environments, which need multiple levels of care that goes into, the research. But yeah, that was very, very interesting. The next question that we have for you is, can these muscle-powered robots be scaled up beyond the micro scale?
Yeah, again, a great question. So I would say that right now they're probably at the millimeter to centimeter scale. The thing that is preventing me from making them bigger right now is that I haven't engineered a blood vessel supply or something similar throughout the tissue. So the way that all of the cells inside this construct, pretend this is a millimeter, are getting in is that they're diffusing in from the surrounding liquid media. And if it got bigger, eventually you hit a diffusion limitation, right? Like, so all of the cells in the middle of the construct are going to die because they're starved. They're starved of oxygen, they're starved of food, they're starved of sugar and amino acids. So they can't live. So if I wanted to make them bigger, I would need to engineer some sort of blood vessel supply such that all of this liquid media is being perfused throughout the tissue so you don't have this diffusion limitation. So that is absolutely something that I and a lot of other people in this field are working on. It requires new advances in 3D printing. It requires new understanding of how to generate branched vasculature or blood vessel networks within tissues, which is interesting not only for skeletal muscle, but any other kinds of tissues that people are engineering in the world. So it's definitely something that the field is working on, and it's something very important. One thing that I think for me is at this scale, I can still ask and answer questions about muscle that are relevant to human biology and to disease. It's not so small that I can't see how thousands of muscle fibers are working together, but it's also not so big that I'm just kind of wasting reagents for no reason, because I can answer all the questions that I have with this scale. So at this scale, I still feel like I can answer the questions that I'm asking. Eventually, if I wanted to say replace somebody's arm because it got blown off in a bomb, then absolutely we would need to learn how to make something that was a lot bigger.
Yeah, that gives us a lot to think about. Obviously, this research in particular has a lot of it has a lot of possibilities. I mean, if it actually works out in the human context, it can be very, very useful. But and its applications are also, there are so many wide applications that it could be used for. So here's hoping that all of it materializes. I think we've received so many questions. We're just going to have to take the last two, I think. So one question that we have here is, how would the body react to biorobots from the perspective of the immune system?
Yeah, fantastic question. So I would never, for example, take the robots that we're making right now and put them inside a human being. And the very obvious reason is that they are made out of mouse cells. So the thing that we need to remember about the immune system, right, is that it's recognizing things as foreign and then fighting against them. So if I were, things that I work on right now are how can we implant these inside mice and study their immune response. And if they are, then we look at how can we change the proteins or the materials that we're making these robots with, and make sure that they're derived perhaps from the animal in which they are being implanted. And this is something that not just us, but anyone who's working on cell therapy or any kind of biological material being implanted to a person is thinking about. And I think that's great because just like with the vasculature, it's one person can never answer all of these questions. So you need the field as a whole working on it. And I think that there are other researchers right now who have talked about how we can go from human skin cells to induced pluripotent stem cells, which can then be differentiated into skeletal muscle. So because of that work, we can then combine that with our work and perhaps fuse these things together where we replace our mouse cells with the human cells and then make constructs that should be made out of the same cells that compose the human being from which they were harvested. And then perhaps their immune system won't have as strong of a response to them. It still might, we don't know. And maybe we'll need to, give them therapeutic drugs that help them fight that response, or perhaps coat the muscle with something so the body doesn't necessarily recognize it as foreign. All of those things might happen, and we'll have to address them when they come. But the first thing that I would do is to move from mouse cells to human cells.
Definitely. Dr. Raman, I must say that the zest with which you speak about your research is actually very inspiring, and I'm so sure many of our attendees are getting motivated to actually pursue research in the long term. I think that brings me to my last question for you. Just one second. Yeah. So we have read a lot about the outreach work that you've been doing throughout the course of your research, and we've also read about how much you advocate the presence of representation and women in STEM. You've been working a lot on that in that ecosystem too, and we just wanted to ask you, now, from the perspective of a researcher, how important do you think it is to have platforms such as India Science Festival, where researchers get to engage with public in order to bring science closer to society? What is your take on?
Yeah, that's a great question, and I think really ties into what we were talking about through all the questions. Is there are so many questions that remain to be answered just in this specific field, and when you broaden that out into other aspects of medical research, like cancer research or COVID vaccines or energy or new ways of producing food or agriculture, you can see that if in 2050 there are 10 billion humans that are all dealing with all of these problems, we're going to need as many people developing solutions to address those problems as possible. And to me, when you exclude, in the case of women, if you exclude half of the people that could be working on this problem, or in the case of other underrepresented minorities in science, people of different races or different economic backgrounds, you're just fighting against yourself, really. We're all fighting against ourselves if we aren't deploying the full force, the full strength of human innovation by stopping people from achieving in these fields. And so that's why it's really important to me. One, just because I think it's just and fair to let everybody work on something that matters to them, and we shouldn't prevent them from education. But also, I think even if you were being completely selfish and you're like, I don't care about other human beings, I don't care about other women, you could still be like, but I still want something in 50 years that looks like a safe and happy and healthy world to live in. In that case, even from a purely selfish perspective, you should be saying, let's just throw as many people at this problem as possible so that we can have solutions that help us out. So I think that's why it's really important for me whenever I have an opportunity to talk to young people who are maybe considering science or maybe think it's too hard or not interesting enough. It's a very rewarding career. It's very exciting. And I think it's something that everyone can learn how to do. And so that makes it very special to me. And I hope that this and some of the other talks that you've heard have convinced you that there is some problem out there that is worth you spending your time learning about and working on.
Doctor, thank you so much for joining us. It was such a pleasure having you on board, and it was a very, very interesting talk. And I would like, on behalf of the entire team at ISF, thank you so much for patiently answering all questions posed by our attendees. It was such a pleasure having you on board.
Of course. Thank you for having me. And thank you, everybody, for your questions and your attention.